So we added 5 milliliters of liver substrate into the clear tube, and we added 5 milliliters of hydrogen peroxide, 1.5%, into the red tube, which is the enzyme. We added both into the test tube in order to create chemical reaction. We filled the cylinder all full of water, and then we put a tube and we shook the test tube in order to create the chemical reaction, and the products caused there to be
the product air.
The product air is 20 millimeters.
Yeah, and it ended up in the cylinder, and the product of water remained in the test tube. The lab that we're doing is called an enzyme reaction lab. We're testing for the substrate concentration through serial division. First, we will add 5 milliliters of liver at 1% concentration and combine it with 5 milliliters of hydrogen peroxide, H2O2. The reaction then will cause air and water as products. We will measure the air product as it passes through the tube up into the cylinder. Every 20 seconds, we will measure the amount of air up in the cylinder. We now added the hydrogen peroxide to the liver, and we are shaking it and seeing the air go up into the cylinder.
See that?
After 20 seconds, there was no product of air in the cylinder, but at the time check of 40 seconds, there's a total of 5 milliliters of air at the top of the cylinder. In this tube, we have now added 5 milliliters of hydrogen peroxide and 5 milliliters of water. We will take 5 milliliters of this combination and add it into this tube. We will then take 5 milliliters of the liver, suspended in water, and test the reaction. Every 20 seconds, we will see how much of the air product will add in the cylinder. This test has a 0.5 concentration of liver and a 0.75 concentration of hydrogen peroxide. Now we are doing the second trial, which has 5 milliliters of hydrogen peroxide mixed with water from the mixture that we had before, and we are now going to combine it with the 5 milliliters of the liver. The water and hydrogen peroxide have a 0.75 concentration level.
Oh wait, it's slipping out.
Oh.
Okay, it's fine.
Somebody hold it. There, if you want to.
Just keep going.
Now the bubbles are going up through the cylinder from the reaction, and we're going to see after 20 seconds how much air has been created. We are now doing our third trial, which has the 5 milliliters of mixture of water and H2O2, hydrogen peroxide. The concentration of the hydrogen peroxide is 0.375. We're going to combine it with 5 milliliters of the liver and see the reaction. This is now our fourth test. We have 5 milliliters of H2O2 and water combined. We're going to add them and see how they react with 5 milliliters of the liver. The concentration of the water in H2O2 is 0.175%.